Hello everyone. So in this video we will discuss the question number 33 of GATE 2018. So the question they are asking, which one of the following statement is true with regard to processing and presentation of protein antigens. So they basically wants to compare between the class 1 and class 2 MHC pathway, which is responsible for antigen processing and actual antigen presentation. So let's first see and compare the antigen processing and presentation pathway by the class 1 MHC and class 2 MHC. So in the left side, the pathway is exogenous pathway, which is mediated by the class 1 MHC. So how does it start? It starts with endogenous antigen which presents in the cytosol. So this endogenous antigens, first it will be degraded by the proteasomal pathway and it will form the short peptides from this whole antigen. So now this antigenic peptide, now it will be transported into the rough endoplasmic reticulum via this TAP. So this after getting inside of the rough endoplasmic reticulum through this TAP, this antigenic peptides will come into the lumen of the rough endoplasmic reticulum. After that, this blue one is the class 1 MHC protein. So first this class 1 MHC alpha chain will bind to the calnexin, right? So this is the calnexin. So in the first step, class 1 MHC protein will be bound to the calnexin and then beta 2 microglobulin, right? So after that, calnexin will be dissociated. So from the MHC class 1, here there is no calnexin in this diagram. So after the dissociation of the calnexin, calreticulin and tapacin will bind. So this is the tapacin and this is calreticulin. So now MHC will be bind to the beta 2 microglobulin, calreticulum and tapacin. And in this condition only, MHC can capture the antigenic peptide which was transported into the rough endoplasmic reticulum, right? So only in this configuration. When it is bound with the calnexin, it can't. So first calnexin has to be dissociated, and after the dissociation, MHC class 1 has to bind with the tapacin, beta 2 microglobulin and calreticulum. So after capturing the antigenic peptides, it will be transported to the Golgi complex by this vesicle. So this vesicle, which contains only the MHC class 1 protein and the antigenic peptide, it will be budded off, and before this budding or before the transport of this MHC class 1 along with the peptide to the Golgi complex, so before this thing, first the chaperones should be dissociated. So here the chaperones are calreticulum and tapacin. So before this transportation from ER to Golgi, this tapacin and calreticulum, they will be dissociated. So only the MHC class 1, beta 2 microglobulin and the antigenic peptide will be there. They will be transported to the Golgi complex through this ER Golgi vesicle. So after coming into the Golgi complex, this class 1 MHC and this antigenic peptide, this combination, so they will first come into the cis Golgi, then medial Golgi, then trans Golgi. After that, from the trans Golgi network or TGN, this this complex, which is made up of class 1 MHC protein and the antigenic peptide, they will be transported to the plasma membrane, right? So in the plasma membrane, after transporting from the Golgi complex to the plasma membrane, this class 1 MHC antigenic this class 1 MHC will present the antigenic peptide which was captured. So this is the pathway called as class 1 MHC or endogenous pathway. In the right panel, the exogenous pathway which is mediated by class 2 MHC is described. So in this pathway, first the class 2 MHC alpha and beta bind with the invariant chain. This is the invariant chain which actually blocks the binding of endogenous antigen. So as it is only specific for the exogenous antigen, so so for that reason, when it is in the ER, then you can see in the from the class 1 pathway that if there is any endogenous antigen in the cytosol, they can be entered into the lumen of the ER, and after entering into the ER, they can bind to the MHC. But to block this process, so that it can only bind with the MHC class 1 but not with the MHC class 2. So to make this process specific for class 1 but not for MHC class 2, there is an invariant chain in the ER which will block the binding of the any endogenous antigen and MHC or any kind of, so there should not be any binding of antigen and MHC class 2 when the MHC class 2 is in the ER. So after that, MHC class 2 and invariant chain complex, it will be transported to the Golgi through this ER Golgi vesicles. So from ER to Golgi, this transport will be happened. So now in the Golgi there is MHC class 2 and invariant chain. So in so after that, from Golgi it will be transported to the endocytic pathway compartments or endosomes. So in this endocytic pathway compartments, this invariant chain will be degraded, and it will only leave a small fragment of the invariant chain which is known as clip or cliff fragment. So after the degradation of the invariant chain, the MHC class 2 protein will only bind to the small fragment of the invariant chain which is known as clip. So after this process, when the exogenous antigen will be taken up through the endocytosis process, then it will be degraded to form the small peptide from this whole antigen, and it will be routed to the endocytic pathway compartment where the MHC class 2 is bound with the clip. So after, when this exogenous antigen will be taken by the endocytosis and it will be routed to the endocytic pathway compartment, this so when this exogenous antigenic peptide will come to the endocytic pathway compartments, then there will be an exchange of clip and the antigenic peptide. So clip will remove and the antigenic peptides will be captured by the MHC class 2 protein. After that, this MHC class 2 and which is bound to the antigenic peptide, they will be transported to the plasma membrane where it will so in the plasma membrane, basically this MHC class 2 will present this antigen to the other immune cells. So this is the brief and basic differences between endogenous pathway and exogenous pathway. Endogenous is mediated by the class 1 and exogenous pathway is mediated by the class 2 MHC. Now let's read the options and see which option is the correct. So first option, in the MHC class 2 so in the class 2 MHC pathway, protein antigens in the cytosol are processed by the proteosomes. Is it true? No. Why? Because in the class 2 so this is actually true for the class 1 MHC pathway. In the class 1 MHC pathway, the endogenous antigen which is present in the cytosol, that will be degraded by the proteasomal system and it will be imported to the rough endoplasmic reticulum. So that occurs in the case of the endogenous pathway or class 1 MHC. So this option is not true. In the class 1 MHC pathway, extracellular protein antigens are endocytosed into vesicles and processed. This, and this is again not true because uh, because the endocytosis of the foreign antigen or the exogenous antigen and its processing that occurs in the class 2 MHC pathway. Here the exogenous antigens will be imported into the cell by endocytosis and it will be chopped off. So yeah, so this thing that extracellular protein antigens are endocytosed into vesicles and process that occurs for class 2 MHC pathway, not for the class 1 MHC pathway. So option B is also wrong. Option C, in the class 1 MHC pathway, transporter associated antigen processing or TAP protein is required for translocating the processed peptides generated in the cytosol. It is correct because in the class 1 MHC pathway, you can see when this endogenous antigen will be processed by the proteasome, so these antigenic peptides are translocated into the rough endoplasmic reticulum from the cytosol. So it will basically so the translocation happens from cytosol to the endoplasmic reticulum, and this import of the cytosolic antigenic peptide to the ER, that import is done by the TAP protein, which is embedded in the endoplasmic reticulum membrane. So so in the class 1 MHC pathway, the TAP protein is required for translocation of processed peptides generated in the cytosol. This is the correct option. So C is correct. Option D, invariant chain in endoplasmic reticulum is involved in the transport of peptide and loading of class 1 MHC. Again, it is the case of class 2 because in the class 2 MHC, so because in the class 2 MHC pathway, MHC class 2 is bound with the invariant chain. After that, the invariant chain will be degraded, and in the endocytic vesicle, this this small fragment of the invariant chain, which is known as clip, that will be exchanged to the exogenous antigenic peptides. So that that part belongs from the class 2 MHC pathway. So invariant chain in ER is involved in the transport of the peptides and loading of class 1 MHC. That is not true. So option C is the correct option here.